you, there are more bacteria cells in your body than actual human cells. According to the National Center for Biotechnology Information, this is true. The typical adult human body contains about 30 trillion human cells and about 38 trillion bacteria cells. Speaking of bacteria, when I was little, my mom would always tell me to put the lid down before I flushed the toilet to keep what's in the toilet. And also to put a cover on my toothbrush, always. Recently, when I was shopping for a cover, I noticed that there were different types of covers. So this led me to wonder if they did the same thing or if they had different ingredients. So, does protecting your toothbrush with a cover keep it clean? If so, which cover works the best? Amazingly, to my surprise, after reading the packaging, they all have different ingredients. After some research, I learned that your toothbrush is loaded with bacteria. In fact, uncovered toothbrushes can harbor 100 million bacteria. So what even is bacteria? Bacteria is a microscopic single-cell organism which uses a process called binary fission, which it uses to duplicate itself to form two identical daughter cells until it forms a massive colony. Just so you know how fast these grow, in 7 hours, one single cell can grow up to 2 million cells. And in just 1 more hour, it goes up to 16 million. Moist, warm, dark areas are breeding grounds for bacteria. So also when I was doing my research, I wanted to see what the different ingredients in the toothbrush covers did. So the first ingredient is thymol, which comes from the herb thyme and has been used in medicine for centuries. Then in the brush pod, there's menthol, which is just peppermint, is used in some cigarettes, and lavender, which is oil and is also antibacterial. Then Luminant has silver as its active ingredient and is well documented antibacterial. My hypothesis is that the brush pod would be the most effective toothbrush cover because it has 2 active ingredients, menthol and lavender, instead of just 1, like the other ones. So when I was doing my general procedure, I exposed 4 toothbrushes with 3 different covers into a typical bathroom environment for 3 weeks, and I compared the presence of bacteria in the petri dishes. I knew bacteria needed a moist environment to grow, so I simulated the act of brushing for all my toothbrushes by rinsing 5 seconds to wet, weighing 1 gram of toothpaste on the toothbrush, brushing side to side on a gloved hand for 30 seconds, then rinse the head of the toothbrush for 30 seconds. I did this for 6 days for approximately 3 weeks. Next, I swabbed each toothbrush to collect a sample for the petri dish. I labeled the dishes and incubated them in a dark closet for 7 days to allow them to grow. When I was getting my results, I had a couple mishaps. So the first thing was when we were swabbing the petri dishes, we forgot to moisten the tip, so we were swabbing dry swabs on the toothbrushes, thinking I was getting results. But then we had to order new dishes, and I got it right the next time. Another mistake we had was we had an app called One Petri, and it would give us like wildly different results for the same picture, and we just thought that wasn't going to work for us. So finally we found a good idea, and we counted the number of colonies in each petri dish and did the surface area, which is the area of a circle equals pi times the radius squared, to find each surface area for the different covers. So as you can see, Steripod did the best and the control did the worst. Unfortunately, we could do the area of a circle because most of the colonies are circles. So the Steripod did a whopping 1,858.63% less bacteria than the control. In conclusion, my hypothesis was that the brush pod would be the most effective, but my results do not support my hypothesis. The worst was definitely the control, second worst was the Luminant, second best was the brush pod, and the best was definitely the Steripod. So my biggest takeaways are using the scientific method is reliable when conducting science experiments, and diligence is definitely needed to get the job done, and most importantly, most definitely, Take cover.
Good job. Awesome.
Sandy?
So you covered the toothbrushes while they were still wet, right after you're done brushing your teeth?
Yes.
And you tested the toothbrushes when after that?
Not right after, but yes.
Immediately afterwards?
No, because then we had to incubate them to get the bacteria.
Okay.
How many weeks did it take you to do?
The whole experiment? Probably 2-3 months. Jim?
What happened? What did you do with the toothbrushes?
After? Well, they're right here, so we're probably just going to dispose them. Miss Ashton?
What did you say is the main ingredient of the one that was most successful?
The brush pot, the Steripod had Timol. Jen?
Do you have any theories? We're a house divided, so I'm looking for some ammo here, Ben. Do you have any theories for why the brush that wasn't covered was the worst?
Probably because the bacteria was just getting straight on the toothbrush. But also the covers might have, the covers could have done worse because it does form an enclosed dark space, so it could grow bacteria. Noah?
Could possible mold in your bathroom have affected the toothbrushes?
It could have. We were testing it to all bacteria in the bathroom, like sinks, toilets, showers, everything that's going on in your bathroom.
What would you do differently if you were to do this again?
Definitely use a microscope to see some of the non-visible colonies, and maybe try some different covers.
You want to take one more question, Ben?
I'm sorry, what? Oh, no, we didn't.
Let's go.
Great job, Ben.